Shrimp. Let's do this. We all know them. We all love them. The bad boys of the sea. So I did have plans to make this much earlier, to be fair, but as I was doing the research, I learned the most horrible news, and so I needed to take some time. Obviously, as you can see, what we are talking about today is shrimp and their incredible vision. That was what I was so excited to talk about today. It wasn't until I actually started the research and read this paper when tragedy struck. I'm just gonna rip the band-aid off now so that you and I together can take the rest of this video to process the hurt that we have been given. Because you see these colors in all of their beauty? That's what the shrimp see too. Maybe not even all of that, because, and I hate to tell you this, the forbidden shrimp colors aren't real. But what are the forbidden shrimp colors, you ask? So if you think about eyes and how we see color, humans, we've got 3 cones in our eyes. For red, blue, and green. No, red, blue, and yellow. I forgot the primary colors. These are what help us see colors, etc. If you have damaged cones or less cones in your eyes, you don't see colors as well. The mantis shrimp, however, have up to 12 different types of cones in their eyes. And so obviously the thought there is they can see forbidden colors. What are they? Base boosted blue, red, but this time it's green, yellow, 2. These and more are what were thought to be as the forbidden shrimp colors. But then comes along Hintho et al. From 2014 to burst all of our bubbles. This study was conducted to examine the excessive construction of these bad boys' eyes and see if they do truly hold the secret key to the DLC colors. But first, some shrimp background, if you will. So the shrimp used in this study are Heptasquilla trispinosa. They're in the order Stomatopoda. They're generalist carnivores, which means they mainly eat small crustaceans, gastropods, macroplankton, plankton. They're generally under 35 millimeters in length, which means that they're very small, but they really pack a lot of power in their punch. And boy howdy, what a punch it is. This specific type of mantis squid is known as a smasher, which means that they will lie in wait, and then they will just smack the living daylights out of an unsuspecting piece of prey. You know the age-old question. Since impact can generate heat, how many slaps does it take to cook a chicken? So I did the math. It would take approximately 72,134.5 of my own personal slaps to cook a whole chicken. I spent a whole day on this. I didn't need to do that. What these kids do is that they move their claws so impossibly fast and transfer so much power into their prey that they one-hit KO basically anything. It's insane. Now I haven't done the math for them yet because I didn't find a way to get the mass of one of their arms, but it's safe to say they can cook a chicken in significantly less slaps than someone like me. But unfortunately, we aren't here to talk about shrimp and their incredibly powerful arms. We're here to talk about their eyes. So if you imagine a shrimp eyeball, basically they're split into 3 main parts. You've got the top part, the bottom part, and it's wearing a fancy belt around the middle. Kind of like our own Earth, if you think of northern, southern hemisphere, and then the equator is the belt. So in their Gucci belt, they've got a whole bunch of different photoreceptors and support cells, and these are divided into 6 main rows. Rows 1 through 4 deal with color processing, and then 5 and 6 deal with detecting polarized light. So within those rows 1 through 4 is where the 12 different types of cells are that differentiate color, as opposed to, again, our 3 human ones. And since these shrimp have such base-boosted eyes, this study was looking at whether they are actually doing different things than humans. So it all said, how do they process these colors? So 1, are they using multiple dichromatic color opposing systems to determine color? Or 2, are they dumping these into 12 different channels without comparing? So, to see which of these hypotheses is correct, the shrimp were tested on their ability to determine between different hues because that correlates to your ability to distinguish between colors. The shrimp were trained on food reward to distinguish and recognize a handful of different colors. The shrimp were then shown the colors that they had been trained to recognize and then a different hue, and then had to distinguish between the 2 of them. When they were shown colors that differed by 50 to 100 nanometers, they were able to distinguish the color. Say that as a nice, gentle powder blue, as opposed to this horrible green. They were able to distinguish this about 80% of the time. You go, you funky little shrimp. However, when the shrimp were shown colors that only differentiated by about 25 nanometers, say this nice gentle blue and this slightly less gentle blue, they they were only able to distinguish the difference in colors about 50% of the time, almost as if they had been guessing. That's nothing. The average human who's able to see without significant color or otherwise damage would be able to distinguish between these two colors. These dang shrimp aren't even using their base boosted eye for any good. What's the point? I need a minute. So let's take a look at some figures to help us figure all this out. Alright, so if we take a look at our first figure here, we're showing on our x-axis, you've got the wavelength in nanometers, and then the y-axis is the normalized sensitivities. And then, of course, you can see each of the colors are how sensitive these eyes are to all of these different wavelengths. Looking at figure 1b, you can see an up-close image of this eye. You've got, as you can see, labeled dorsal hemisphere, ventral hemisphere, and then in the middle, that wonderful, gorgeous belt. And then in that gorgeous Gucci belt mid-band, you've got the first 4 rows being where the color receptors are, labeled by the CV. Gorgeous, incredible, love to see it. Next, we've got A, B, C, and D in figure 2. This is essentially showing the success rate of these different tests that are happening. So A and B, you've got success rate percentage on your y-axis, and then at x-axis, you've got the separation between your stimuli, again, in nanometers. And you can see as the separation between the stimuli increases, the success rate also increases, showing that the farther apart two colors are, the easier it is for the shrimp to tell the difference. Makes sense. And then you can see over here, C and D, you've got, again, success rate percentage on the y-axis and separation between the stimuli on the x-axis. With part C, you can see that as the wavelengths get shorter and closer together, the success rate just plummets. And clearly, these shrimp are having a little bit of trouble with their colors. Maybe they, like me, need to go back to grade school and remember at least their primary colors. You can also see the number above each point, which indicates the tested wavelengths. Figure 3, we've got spectral discrimination curves with change in wavelength in nanometers on our y-axis, and on our x-axis we have the wavelength in nanometers. And this is showing a handful of different organisms. The dark thick line is our stomatopod, and then down towards the bottom is the mottled system. And this shows us the sensitivity for all of these organisms to different wavelengths. And finally, figure 4, we have the proposed processing mechanism. Part A is giving us the normalized reflectance from the stomatopod itself, showing the wavelength, again, on our x-axis, and the normalized percentage on our y-axis. Part B illustrates more effectively one of the hypotheses that the shrimp are actually binning each color into separate sections without comparison between the two. And then C and D are showing the blue and red stimulus respectively, and the excitation patterns that are then brought to the brain itself. But anyway, that's just a very long-winded explanation. Let's get to the real stuff. What do all of these mean? What did the scientists conclude? Well, I'll tell ya. So overall, the scientists conclude that these shrimp use their excessive number of cones in their eyes to scan the sea, and they're able to detect predator or prey by color with lightning-fast reflexes. So while these boys do have excellent color vision, it's just not as cool as we had hoped it was. But, you know, maybe out there somewhere, the shrimp colors do exist. For those of us who believe, maybe they're up there in shrimp heaven.